The next speaker is Dr. Govind Ragupathi. He is a senior immunologist at Memorial Sloan Kettering Cancer Center, and he has been involved in the development of cancer vaccines since the early 90s. He is a recognized expert in methods for conjugating different antigens to carrier molecules. So he will be talking about botanical immunoadjuvants for cancer vaccines.
So now I'm finding where my topic is fitting, because always I'm working on the defined molecule, and now most of the talk here is whole extract and everything. So that's why my talk going to go back and forth with adjuvant, natural adjuvant, and synthetic vaccines. So here my topic is botanical and synthetic immunoadjuvants in cancer vaccines. So my expertise is mostly on cancer vaccines. So years ago, we identified many antigens which is expressed on cancer. So some of them are already I mentioned, the glycolipids and glycoproteins. And the antigen which is the glycolipids has advantage than glycoproteins. So which is entirely different topic on the immunology, why these antigens are important. For example, we screen the tumor samples by micro tissue array, all these things using the monoclonal antibodies. We identified melanoma, for example, expressing GM2, GD2, GD3, and neuroblastoma. Most of the antigens are sharing with it's a common antigens. For ganglioside, which is common, and you have STN, TN, TF, mucin, all these things. And also we have for example, when you go to the prostate cancer, you don't see a GD2, GD3 expressing on the prostate cancer. Come to globo H and cellular Lewis Y and TN, TF, all these glycoproteins. How this molecule present on the cell surface? So, for example, GD2, GD3 are all on the cell surface, and for other glycolipids, which is the glycoproteins, which is expressed on cellular Lewis A, globo H, which is entirely different class of molecules. Our aim is here to develop antibody to bind on this molecule, and this antibody induces the complement, all these things. That's a therapeutic purpose. So in the morning I present this slide. So how to induce the antibodies. This is a concept, regular concept. So conjugate with the antigen, conjugate with the carrier molecules, and add some adjuvant. So the adjuvant is a lot of adjuvant we can use. The best known people are using on infectious disease is alum. So what we are start using with the QS21, which came from Coila sapanoria. This is a commonly available plant in South America. So 20-30 years ago, Dante Martian said, oh, these are the ones we are using an adjuvant in the plant extract. So he identified that, he fractioned that, he wants to see exactly what active principle is presented. So he named the compound. Whatever comes out is based upon his fraction, HPLC fraction, QS21, 7, 10 different names. And also I want to point out that the QS21, the extract is also used in Coke. It's like a soapy solution. So I don't know what the purpose of Coke company is using. So this plant is very highly demand. It's very difficult to get a natural antigen. So then you have Dante Martiani group isolated the QS21, and they contacted my Sloan Kettering Cancer Center to test that in the cancer vaccine along with our vaccines. So what we did, we picked the one antigen called the GD3, which is abundantly expressed on melanoma. So the reason, then we conjugated with KLH and just compared with this adjuvant QS21 with a different commonly available that time, that adjuvant. So what we have seen is QS21 is the superior, and also titer max also is a superior one, but somehow the QS21 we are able to get few and also enough quantity to do the clinical trial. That's why we set on QS21. So after this preclinical, we did a lot of characterization, just to this slide, not only adjuvant effect, antibody, complement, facts, everything we characterize, which takes another talk. So then we took into the clinic, we put that thing. So I'm going to go back and forth the different antigen because because we did clinical trial almost all the antigen which I showed in the previous slide. So this is the QS21 comparing inpatient with GM2 vaccine, which is all my vaccine here I'm talking is conjugated with KLH, the one gold standard. And we happen to see that QS21 is stood on the top, which is able to induce IgM and IgG response. So you can clearly see the titer. But the dose which is we selected that time, 200 microgram, is toxic, which induces erythema in the patient. So that QS21 we can use in the cancer patient, but we cannot use in the kids as infectious disease vaccine because your arm going to be swelling like big. So then we adopted a similar approach of almost lot of antigen and tried in a different cancers. For example, GM2 we tried in melanoma, GD2 melanoma. For fucosylated GM1, which is another antigen very specific in small cell lung cancer and neuroblastoma, global which is specific in prostate and breast cancer. We did all this. One slide summarizes many years of the work done by both clinician and basic scientists in Sloan Kettering. So then we said, okay, what happened? We have the antigen. So what happened? The company which providing the QS21, they're giving it trouble because of intellectual properties, all this came. So this is a brief history of what the QS21 came. The reason is, we are all working the plant product. How long it takes from the extract to do the clinical trial and identifying active molecules. So this is a very good summary of QS21. For example, in 1986, they identified a QS21 fraction is immunologically active, adjuvant active, and 1992 and 93, Sloan Kettering started the first clinical trial. Then slowly it goes. So this is the summary, and the patent expired. And then we are also desperately looking for a new adjuvants. So we collected whatever the based on literature, we contacted many companies, many scientists. We gathered about 26 different adjuvant and tested. Again, for the interest, you see that still the QS21 is the top of our list, which is inducing both IgG and IgM response and the same antigen. And GPI 100, which is same thing like a Dante Marchionne when he isolated and then he went back, he's a chemist. So you want to make the molecule synthetically potent molecule, that's what he called the GPI 100. So GPI 100, based on this result, we took the GPI on clinic. The dose is very high compared to the QS21. QS21, all we need is a 20 microgram. GPI, you see, we need almost like a 1000 microgram. Whatever it's a QS21 is 100 microgram patient. So this is again, what happening is we also, instead of monovalent, we are also tried with a polyvalent. Polyvalent is just, for example, breast cancer vaccine, which is a combination of different antigen together. So here on the first 3 patients, we tested with the GPI 100. But even though the titer looks very high compared to on the bottom, you see that QS21 GPI, the synthetic version, looks the titer is high. But the patients, we see a liver toxicity in the patient, and FDA told us we cannot continue the trial. So still we gone back to the QS21. So the company which they're providing the QS21, they're giving the trouble. So whether we need to make our own QS21, or maybe we want to synthetically make the QS21. So Sloan Kettering hired a chemist, especially for this purpose. So his name is David Jin. He took the synthetic part of the QS21, and we contacted the other company. They synthesized, they purified from the plant source with RP21. We called RP21, which is a synthetic one. So the QS21, it tests like a chemist always has a different opinion. Oh, Dr. Raghu, if you make the QS21 is an isomer, which is naturally present, is a 65 and 35 ratio. They said, okay, even if you make one molecule which is abundant, that's a 65% molecule, and we don't need this one. Otherwise, we can do the combination. All this chemistry guy came with all this proportion. So here I can say that QS21 has a 4 quadrant. One is a trisaccharide, tetraterpene, tetrasaccharide. One is acyl chain. chemistry group, they synthesize this and they try to cut off, because QS21 is toxic. They want to know which is the less toxic. So they modified all these things, which is coming back. And the first step, they made a QS21 synthetically, which is APOs and xylose combination. And it involves the 62 step, which took a long time, maybe like a couple of years, to Dr. David Jin group, chemically achieve this molecule, which is very complex molecule. And then he compared with everything with what is naturally available, synthetic, everything. He theoretical compare. Oh, this is almost same as natural molecule. Then we tested, my lab tested on the preclinical studies. So we saw that it's very good. Same again. Always I try to use the same antigen to compare the outcome of the previous results. So here I used a GD2, which is again present in melanoma, sarcoma, and neuroblastoma. And again, the QS21 with the APS alone, xylose alone, mixed with a 65-35 ratio, which we called SQS21, called synthetic QS21, and GPI 100, which I told you previously, it is a toxic. But still, surprisingly, GPI 100 showed very good immunological activities, but we cannot continue. We did a trial in melanoma comparing the natural QS21. Then this is the summary, and again here we can say, okay, different form of the QS21. Okay, it induces very high titer of IgG and Ig response. Those antibodies are reacting with melanoma cell lines. Very good. So we got it very good results. Then the second step is taking this to toxicity, same toxicity which we saw observed in the QS21. Always the QS21 is toxic. So then we combine these two antigens together for treating a melanoma patient, one GD2 and GD3. So this is the vaccination protocol in patient. So that trial compared side by side, which is natural form of the QS21 mixed with the same vaccine and synthetic form of the QS21. The patients are vaccinated at week 1, 2, 3 and week 8 and week 21 and week 32. And also we collected blood over the period of times, and comparing the best one is ELISA. So theoretically, you see that this is just a representative of total 12 patients. So OP21 is the natural one, which is induced a very good IgG response and very IgM response, and it is also induced a very good GD2 response, a good response against GD2, and the QS21, synthetic QS21 also we see that. So still, if you can see this, there is a little bit difference. The compound came isolated naturally, the compound synthesized by chemical method. And then on the program, so previously I told you the QS21 as a 4 quadrants, then we chopped up and converted all these things because we tried to remove the toxicities of this molecule. So we can name it like a SQS1, 101, SQS 102, 103. Again, we screen the same way, again inject to the mice with the vaccine. In this case, GD2, GD2, and previously here is comparing the toxicity. So you see that SQS 102, which is very toxic. Even if it's this much toxic in mice, it is very difficult to take this molecule further. When in the next slide, you see that the antibody response is comparably good with all the molecule, but the selection based upon antibody response, so like a QS21, we see like a IgM titer is close to 600, and the IgG response QS21 is very good response. But we made a similar, like this kind of all the molecule just cutting off and changing the portion, putting acyl group, removing tetrapaine, all these things. Total David Jin group made about 80 molecules with various combinations. And we identified a 1 or 2 molecules which is very, very immunologically active, is better than a QS21, which is licensed out to the company for further development with the cancer vaccines and infectious disease vaccines. So then the natural product, the program, we naturally isolated QS21 from the plant product, which we called as OP21. And the OP21 as exactly, once you make the molecule, you need to compare with the old existing literatures, and NMR, the OP21 which naturally isolated is fit with the synthetic profile and everything. So we are happy with the new molecule. And since the QS21 synthesized synthetic QS21 is less potent than the natural one, we decided to take the natural QS21, which is called OP21, to further clinics. So what we did in a second phase 2 trial, we picked the 3 trivalent component, which is GD2, GD3 and GM2 mixed with the vaccine. We did a trial. The trial is ongoing, and also the same thing with the ovarian cancer combination heptavalent, which is natural. OP21 is ongoing. So there is another trial which is ongoing, which is very interesting, is the same GD2, GD3 combined. We call it the bivalent vaccine mixed with a natural adjuvant, but gavaging with a beta-glucan. So usually we give the vaccine, nothing treated. The beta-glucan is a natural source which is isolated from yeast, and also we have a barley beta-glucan, but the trial here is mostly came from beta-glucan, which is from yeast. So the same vaccination schedule, weekly injection, but after 3rd injection, patient, this is a neuroblastoma patient, kids are given oral beta-glucan. So here we found a very results that neuroblastoma is a deadly disease and its average lifespan is 3 to 5 years. So kids who got this vaccine tend to live longer than the kids who didn't get the vaccine. So with this trial would have researched only 14 patients, and we are continuing the trial, and now about 200 patients have been vaccinated. I don't know the exact outcome of the results. So what I'm going to do for the AYUSH, I got a extract, asparagus extract and Vithoni extract from Grish, Dr. Grish, and I think I showed this the last morning. I found that the Vithoni extract is immunologically more active, which is more superior than my gold standard RP21. So here I'm debating whether we need to test this molecule as individually as an extract, or maybe we can fractionate and see what the active principle is similar to what we have seen in QS21. So that's my next plan going on that. These are the group of people who contributed clinically, because I cannot put all the clinicians who involved with this trial. So David Jin group is the one synthesized all the molecule, and this is from my lab. We did all preclinical and made the vaccine for the clinic.
If you have one quick question from anybody in the audience.
Vithania extract. Vithania is a very difficult ball game. Everybody is playing all over the world, India also. And phytochemistry of Vithania is too complex, and there are chemotypes also, varying chemotypes. So you have a profile of extract you used, and which are the dominant vitellurides, which are all the vithaferin A is how much, etc.
Oh, so here, just I'm putting this one. Actually, I used it's on the dose is on the bottom, 100 microgram. You can see that.
So is it necessary to go for both IgM and IgG titers evaluation, or IgG would only evaluating IgG would also be sufficient?
So both have a different mechanism of actions. I would prefer the one is like IgM is a short-lived antibody. IgG is a long secondary response, all these things. So better for any given molecule, it's better to induce the IgG response. But this molecule, all this one ganglioside, which is a carbohydrate, majority is the primary response. There is no secondary response, IgM. But if you ask me, because I have other immunotherapeutic program, developing antibody, where I'm specifically looking for IgG. IgG 1, 2, 3, all these things.
Thank you. Just one more question related to Vithania, sir. Are we looking for the combined, immunological response of the whole extract, or do you feel that it would be like a synergistic effect, or it may go working more better for individual compounds?
Okay, so in this I'm coming back to a question later. Here, just I was debating whether I want to mix with the QS21, my cocktail, mixing with or administering all these things. In this case, just vaccine, no adjuvant, mixed with Vithania. So I. Yeah. You told me yesterday. So yeah, the profile and everything. So, but I didn't get it. But see, the reason why I did this, I want to see whether is there any activity. If it's not any activity, then I don't want to waste my time. But I was so impressed after seeing this because always my gold standard last 15 years working with QS21 up to 821, when I see this one, Vithania is the good one. And also I got tried with many botanical products from Dr. P.C. Ling from Hong Kong, but I never seen this kind of response.
Okay, thank you, Dr. Ragupathi. Yeah?
Thank you.
Thank you very much.
I now invite Dr. Denis Chang, Professor of Pharmacology and lead of National Institute of Complementary Health, Healthy Hearts Research Program. His area of expertise is clinical and pharmacological studies of Chinese herbal medicine at Nickham and Western Sydney University. Sir will be talking about preclinical PK-PD modeling for herbal drugs.
Thank you, Preeti. Good afternoon, everyone. It's good to come back to Pune in this more welcoming weather because last time I was here it's really hot. So you probably noticed that I have changed my topic a bit because instead of talking purely about the PD-PK model, which I will describe that as well, but I just want to give you an example the kind of work we are doing to develop some novel herbal drugs for the treatment of serious chronic illness. Because we often debate about the role the herbal medicine play, regardless of whether it's a Chinese herbal medicine or Ayurveda herbal medicine. But we have been working with a group of scientists in China over the last 15 years, which is generated the result actually I'm just about to present. We're targeting vascular dementia, which is obviously one of the very serious chronic illness. As we know that in general, dementia has becoming a very devastating health problem in modern society, not only in Western countries, also in around the world as well. So I wouldn't actually go through those figures, but other than pointed out that the cost of the dementia has skyrocketed. For example, the total cost of dementia care over the last 2 years is about $1 trillion per year. Which is, put that into context, you probably don't know the size of Australian economy. Australian GDP is about $1 trillion, which means that if dementia were a country, that is probably will be the 17th or 18th largest economy in the world. That actually gives you a context of the course of dementia. I haven't actually seen any topics in this conference so far because we're all talking about cancers and cardiovascular diseases. And in fact, neurodegenerative disease is becoming a more and more problem in our aging population. So we know that there is a couple of the type of dementia. For example, the most common type is Alzheimer's disease, and which actually followed by the second most common type is vascular dementia. And together with several other less common type like frontotemporal dementia, Lewy body dementia, etc., etc. So what we have been targeting is vascular dementia, which is mainly caused by mini stroke. Obviously, lots of atherosclerosis-related disorders can eventually leading to stroke and especially mini stroke. In many of the vascular dementia patients, they don't even notice that they have this mini stroke because they just gradually losing their neurocognitive function in the more stepwise approach. So at the moment, the vascular dementia constitute about 20 to 30% of all dementia cases. In Asian country, this percentage can be higher because, relatively speaking, Asian country, especially China, India, and some even poor Western, like Asian country, have relatively less well control of cardiovascular risk factors, which means that they develop into a stroke easier and earlier than some Western countries. So that's why the prevalence of vascular dementia is higher in Asian country, or obviously some other relatively less developed regions as well. So I won't talk about the clinical manifestation of that, other than pointing out that at the moment, we haven't got any cure for, we haven't got any cure for any neurodegenerative disease anyway, but in terms of vascular dementia, we haven't got any approved pharmaceutical medicine for the treatment of that. For Alzheimer's disease, yes, we have some Aricept, which is acetylcholinesterase inhibitors, but for vascular dementia, we haven't got any approved pharmaceutical medicine. So that's exactly the reason why we've been targeting this, because we feel that traditional medicine, complementary medicine, might be able to play a role in the areas and that Western pharmaceutical medicine have not got lots of options, especially in the chronic disease management, especially in the prevention of any chronic illness. So that's why we're targeting vascular dementia. Obviously, this shows the therapeutic targets of vascular dementia. I won't go through them in details because of the time limit. But other than pointing out that this is a very complex process due to ischemia, which is leading to glutamate-induced neurotoxicity, calcium overloading, apoptosis, and exaggerated by oxidative stress and inflammation, and so on and so forth. So when you design the drug that actually target these particular units, you have to take into that consideration of all those multiple therapeutic targets. While as we know that pharmaceutical medicine often have a single entity and single target approach, which might not be ideal. So that's why we've been looking into some traditional knowledge, what sort of a herbal medicine, and that actually has some scientific merit, and that can be used for the treatment of vascular dementia. That's how we arrived these particular three herbs. Obviously, Panax ginseng, as you probably know, that is the so-called king of the herbs in Chinese herbal medicine. And ginkgo biloba, obviously we know that ginkgo biloba has been used for the neurocognitive enhancing treatment for many, many years in Europe and in many other countries as well. Saffron, obviously, is the one we actually use for cooking, but we also have some scientific data to suggest that saffron can actually enhance blood circulation to the brain, and also they have antidepressant effect, which is handy because lots of dementia patients do have depression. Okay, so that's why after we finding those three, so that's actually we designed a process. If you're familiar with the pharmaceutical medicine development, and this is quite similar to that. Literally, we have divided into three phases. I mean, the phase one is so-called the formulation development or pharmaceutical development, and where we are doing a whole range of using different extraction method and try to identify what are the active components from each of those three herbs, and try to use a different extraction method to bring out the bioactive component from each of those herbs. And then we combine them. We combine them in a different ratio, 5, 5, 5, 1, 1, 2, 3, 3, 2, 1, and a whole range of 20 or 50 different combination. And then we're using by-assay guided fractionation. We're using different animal models to testing a different ratio and to arrive the optimal ratio among those three herbs. Three herbal extract, I should say. So once the formulation is defined, then we actually develop quality control process as well, where we're using 10 different markers and try to define this particular herbal formulation. And then in the finished product, we know that how many of the biomarkers, bioactive components in it, and how much is in it, etc. So that's the first phase 1. Phase 2, which is a clinical studies where we have done whole range of pharmacokinetic and to work out the mechanism of action behind that, and pharmacokinetics to look at when they appear in the blood, whether they be able to enter into the target organ, which is the brain, and then before we do in clinical trials. In fact, we have done 2 phase 1 clinical trials and 2 phase 2 clinical trials, which I briefly describe the result in a minute. And then currently, we are doing 2 phase 3 clinical trials, so 1 in Australia, 1 in China. So I just selectively show you some of the data we collected so far. For example, in terms of the ratio optimization, so we're using 2 different models. So one is the ischemia reperfusion model, and the other one is Morris water maze model to testing the neurocognition. And based on that, those are 3 individual herbal extracts, and we rank them according to its efficacy in those 2 models. So eventually what happened is that in mice, we found out those 3, the ratio among those 3 herbs are 20-24, and then in rat model, obviously is the ischemia reperfusion model, we found that it's a 15-15-3. So eventually they are quite consistent, surprisingly. So therefore, the final ratio we use in the formulation among those 3 herbs is 5-5-1. It means 5 parts of the ginseng, 5 parts of the ginkgo, and 1 part of saffron. So, and this shows the so-called quality control processes we got in place. As I said that we have identified 10 markers as the quality assurance markers. So basically 4 markers from ginseng, 4 markers from ginkgo, and 2 markers from saffron. So in fact, this is quite unique because so far no drug regulators in any jurisdiction actually require that many markers. So in the quality control process. So we have 10 for that particular herbal formulation. Obviously, we also do pesticide control, heavy metals, and so on and so forth. That's quite routine. So this actually shows you the animal models we have used for the to evaluate the mechanism of action. So we use a whole range of cerebral ischemia model, neurocognitive impairment model, vascular dementia model, and other models. So I won't go through them in details, rather than showing you the extensive models we have used in the team to define the mechanism of action of this particular herbal formula. So I just show you a couple of results. For example, this is the so-called the middle cerebral arterial occlusion model in rats. So we show that basically used to be called WNK, now called SLT. So both medium and high dose compared to control, that actually significantly reduced the infarct area in the brains of the rats. So this is another vascular dementia model actually induced by D-galactulose in mice. So in Morris water maze, we identified that both the median and high dose compared to control has significantly reduced the total length of the travel and also the latency where the mice found their exit out. So which implied that there was a significant improvement in their neurocognition and their memory function, allowing them to find the exit earlier and quickly. And also, interestingly, we also done some in vitro study in the brain of the mice. So to look at the choline esterase concentration. You know choline esterase is the enzyme break down acetylcholine. And we also know that acetylcholine is a very important neurotransmitter for neurocognition. So in doing so, we identified that basically medium and high dose of SLT can significantly inhibit acetylcholine esterase activities, which is a good news because which means that acetylcholine can last longer in the brain and which will help to maintain the neurocognitive function. So to put all those results together, we actually actually produced like a 2 PhD thesis thickness, the result based on those models. But this actually table summarize all those pharmacokinetic mechanisms of action. So this left column is basically shows you the whole range of the therapeutic targets associated with vascular dementia. And actually you can see both ginkgo, ginseng, and saffron individually they different markers, different bioactive components actually target different therapeutic targets associated with the disease, which is a truly so-called multi-component and multi-target approach. Pharmacokinetically, we also done some animal studies as well as human studies. For example, this actually is the rat pharmacokinetic study, shows you individual herbal extract have different bioactive component appear in the blood after the oral ingestion. So you can see that there's many compounds can be found in the blood. Some last as long as 40 to 80 hours, but most of them actually last about 4 to 8 hours, which is a very standard in many of those small molecule pharmacokinetic, which is very consistent with that. We also have done some pharmacokinetics to look at the brain absorption of the bioactive component. Obviously, we show that there are some bioactive components of the formula has in fact can be detected in the brain tissue, which means that that bioactive component have successfully bypassed the blood-brain barrier into the brain, which is the target organ of these particular diseases. And so this is some clinical pharmacokinetic study. In fact, we have found 18 bioactive components can be identified in the human. This is the human pharmacokinetics in the plasma of the human subjects. So based on that, we decided this is good enough, and obviously the whole set of pharmacokinetic and pharmacodynamic shows us that this have enough scientific merit. So that's why we move on to do some clinical studies. In fact, we have done 2 phase 1 studies, which in healthy volunteers, we have done single dose and we have done repeated dose. Obviously, I forgot to mention that in preclinical development, we also done some acute and chronic toxicity study as well. So, which shows that most of the people have no severe adverse event, even though there was 1 or 2 of them actually developed some GI problem or skin problem, but that's very standard because most of the pharmaceutical medicine have that too. So then we move on to do 2 phase 2 studies. So the first one, phase 2 study is basically, we have done that in Australia. So basically we recruited 62 vascular dementia patients, where this is a VDES-COG, which is the primary outcome measure to evaluate the neurocognition. In fact, the 16 weeks treatment, which is 4 months treatment, you can see that there was a significant reduction of the score, which the lower the score, the better, obviously, which means there's improvement in neurocognition. So basically, the net gain of that, the VDES score is about 3 units improvement. So far, all the pharmaceutical medicine, the best they can do is about 2.7, 2.8 units improvement. Even 4 months treatment of SLT has already achieved 3 units improvement. More interestingly, we have done SPECT scan in those patients. We managed to show that there was an increase in the cerebral blood flow in certain part of the brain, including the inferior temporal and anterior temporal region, which is consistent with those brain part responsible for neurocognition. In Chinese study, this is a dose determination phase 2 study, which is a bigger. They recruited 325 vascular dementia patients. They actually do 2 doses, and low dose and high dose, and then they have a placebo control, but only for 6 months, because after the 6 months of a placebo, they randomly allocate them to either high dose or low dose study. which is a quite unique experimental design. So similarly, they use a VDES-COG as the primary outcome measure. You can see that the endpoint of the treatment is 26 weeks, which is a half year. So actually, you can see that both low dose and the high dose have achieved about 4 units improvement in VDES-COG, which is even greater than what we have seen over 4 months. So after that, you can see this is a placebo. After the placebo, after 6 months, they randomize them into either the lower dose or high dose of SLT treatment as well. You can see that the placebo group actually follows similar trend after they switch to SLT treatment, which is a very consistent with the observation we have seen there. Similarly, this is the clinician impression of the changes. It's global changes assessed by very experienced clinician in terms of the neurocognitive function changes. They actually follow very similar patterns, as you can see, the decrease, which means improve in their neurocognitive function compared to placebo control group there. In summary, so this set of experiment actually demonstrate that the feasibility of developing some novel herbal medicine actually that can be used for the treatment of more severe chronic illness. So if successful, we will actually register that with TGA and FDA, and these may well become first approved medicine for the management of vascular dementia. So at the moment, we're actually doing at the final stage of the 2 phase 3 studies. Hopefully, in the next 2 years, we'll be able to finish those phase 3. And if the results are successful, then we'll go ahead with the registration with the drug regulator. Thank you very much for that.
Any question to Dr. Chang?
The formulation involved only bioactive fractions or the plant, material extract?
We purified that. Basically, the 80% of the component are bioactive components. So we managed to eliminate those fibers and carbohydrate and those non-necessary and useless component, make them into a very small capsule. In fact, it's a very much like a pharmaceutical medicine, 2 size 0 capsule twice a day. So it's very, very neat in that sense.
Thank you, sir.
I request Dr. Satyamoorty to please felicitate Dr. Chang. We will have Dr. Satyamoorty's talk on overview of omics technologies for Ayurveda research.
Good afternoon, good evening rather, it's already 4:30. I think I will take a few minutes to review what happened in the session, and then I'll try and finish my proposed talk within the next few minutes. The session opened with the talk by Dr. Bhushan Patwardhan, who made a very poignant statement to say that don't vitiate the basic principles of Ayurveda, but instead integrate Ayurveda into your research practices. And a very poignant statement, I think, which is very apt and appropriate for the type of philosophical thinking which we have in the field of Ayurveda. Dr. Kalpana Joshi and along with Dr. Ramesh Bonde have beautifully expounded the effect of various types of rasayanas, especially to differentiate stem cells, and not only just to a single lineage but to a multiple lineages. And they show some very clear evidence to show that different rasayanas may have different properties on induction of differentiation or proliferation into stem cells. Subsequently, Dr. Shoche spoke about microbiome, especially those related to prakritis and distribution of various type of microbiome, not only coming from the gut but also from skin, which he showed by 18S ribosome sequencing. And he also finds a specific categories of microbiome which are associated with either kapha, pitta or vata prakritis. We had next we had a talk by Dr. Ragupathi who was in fact spoke about adjuvants, especially in the clinical trials, vaccine clinical trials, and also showed a profound effect of Withania extract in being able to increase the antibody response due to vaccine therapy. And lastly, Dr. Denis Chang wondered why people are not concentrating on conditions other than cancer or anything else, but he spoke about the issues of dementia, and then how they were able to prepare the herbal extract to the most purified, to most active forms to a highly concentrated form to be able to get a good effects, as he has shown with a number of parameters which are associated with the cognition in his experiments. If there are any questions to any speakers, you are welcome to ask, and this is the time, because then I will quickly give the talk which I am assigned to. Corridor questions. Okay. Thank you, Dr. Patwardhan. I have been given an unenviable task by Dr. Girish, who is not here, to be able to talk about the omics technologies, which is almost impossible to summarize every single omics technology which is currently being developed or being used, and also talk about the genomic basis of Prakriti, and I was told that I have about 15 minutes. So I'll be skipping quite a bit of slides. Again, we can make it as a corridor talk like Professor Bhushan Patwardhan said, and I'll be happy to talk to you further. So human genome per se is very complex. We seem to realize every day. We did not realize it in the year 2000 when it was sequenced, but now we seem to realize that it is much more complex than it is just a 3 billion base pairs of sequences. And starting with the SNPs, which was thought to be approximately about 1 million of them in our genome, but we see that there are approximately now 8 million of them in our genome. We used to think that genome is 99.9% identical between two individuals. Now we think that genome is 99.2% identical between the two genomes. And I won't be surprised in the coming years that there will be more and more differences which will be found among the individuals in the human genome. Repetitive sequences, although they form nearly about 40% of the sequences within the genome, but they are still complex and they are not very easy to understand, because these repetitive sequences also have a significant role in maintaining the plasticity of the human genome. The copy number variations are very, very important, assuming that 70% of our genome is in fact has variations in the copy numbers within the DNA segment, and that makes it all the more very important that how they are able to provide a kind of a gene dosage effect that regulates the transcripts and subsequently the proteins in being able to bring about various phenotypic effects in a given individual. And then there are epigenetic events such as DNA methylation and chromatin modifications and the microRNAs, which are in fact are all the results of the effects of the DNA, which subsequently translates down into the effects in a given cell or a given tissue type. So quickly, very briefly and quickly, I'll just give you an overview about what are these omics technologies which are currently being used, and probably how these can be used especially in the study that are related to traditional medicine or Ayurveda. The microarrays is one of the oldest technology which has been first developed. It was very popular in 1990s and year 2000, where one can sample the entire genome within a given piece of chip or a glass slide or a chip, to be able to be hybridized with the targets, which can be either a single sample or the multiple samples using the various fluorescent technologies, which are either single colors or multiple colors, which is indicated here. So it works. Wherein one can identify differences, the differences between the two sequences. So these differences can be from the expression perspective, these differences can be from the copy number variations, or these differences simply can be from some of the epigenetic mechanisms such as DNA methylations. And this has been quite popular to an extent that most of the SNPs or single nucleotide polymorphism studies were done using a million DNA sequence chips, and genome association studies or Hardy-Weinberg concepts have been derived from the studies which are done from the genome-wide analysis using microarray chips. And there are some examples which I have tried to quote, but I will just skip it because of the lack of time. And now then there are series of other genome analysis which are referred to as 3C, 4C, 5C, high C, and chip-seq, which are in place. Now this in fact addresses a very important question of how nuclear genomes are in fact are organized in a way that they can be different when they are quiescent, when they are senescent, when they are highly replicating, or they are in fact are interacting with other cells or cell types within a given tissue or a given organ. So nuclear architecture primarily determines the state of a given cell or a condition. Nuclear architecture primarily determines how the cell should behave and how the cell should migrate, how the cell should invade, and all of those characteristics are associated with the properties of a given cell. So these nuclear architectures are not linear. They are as you know that the DNA sequences per se are not linear. Approximately about 3 billion base pairs are in fact are inside the nucleus, and you can imagine how they are in fact are packed in the form of either euchromatin or heterochromatin inside the given cell. So they ought to be talking to each other within the nucleus. They need not be talking in a linear fashion, but they are talking in a topological fashion. In other words, a chromosome 1 may be talking to chromosome X or Y, chromosome 19 may be talking to chromosome 2 when they are not in the mitotic stage. So how would you understand the interaction between the various segments of the genome? That would give us some kind of an indication that there is a crosstalk between the various elements within the genome. And these are the kind of experiments which we call it as a 3C, 4C, 5C, high C are the ones which are a progression of the technology which is being developed over a last decade or so. Right, ultimately the people are using high C, in which the nucleus is in fact is fixed in a given state or topological state, and then they are subsequently immunoprecipitated, and then they are broken down and then religated again, and it is indicated in the figures, and then they are subsequently sequenced. And then these will tell us the analysis of the sequence which is done would tell us what are the topological events which are associated with the DNA sequence interaction, to be able to tell how exactly it works in a 3-dimensional fashion. Next generation sequencing, as most of you or many of you may know, is quite popular these days. It can be used for many purposes. The limit of next generation sequencing is only the limit of what you can think about. So the type of experiments which you can do to be able to get the useful data or useful information from the NGS is probably very well known. And again, starting from doing the whole genome sequencing to exome sequencing to transcriptome sequencing, small RNA sequencing, all of those can be done with the next generation sequencing technologies, where different chemistries are in fact are available depending on the different type of platforms, and these will provide the appropriate information. And there are some beautiful papers which have used NGS to come out with some of these findings or some of these outputs. Transcriptomics has been indispensable part of to be able to see how the transcripts in fact play a role, either for the discovery of a marker or for to be able to understand what is the fate of a given cell, and in relation to the protein expression subsequently in the downstream. And transcriptomic again using next generation sequencing and also using microarrays can be performed, and especially in the field of Ayurveda and the traditional medicine, it can be a very important tool if one wants to get a holistic picture, just like the holistic picture of Ayurveda. If you want to get a holistic picture of transcriptomics, next generation sequencing is the way to do, to be able to connect, to be able to evolve system based biology in relation to the cellular fate or the cellular behavior. There are a couple of platforms which are used for the proteomics, wherein one will be able to map entire set of proteins which are given in a given cell. Although there are about 20,000 genes in a given cell, a cell has a capacity to make more than 150,000 proteins, and then any two cells at a given time do not make 150,000 proteins, and that's what makes two cells different. And all of these can be identified using mass spectroscopy, liquid chromatography coupled mass spectroscopy QTOF type of machines, which can provide you the peptide signatures, which are subsequently analyzed using bioinformatic concepts and bioinformatic principles to identify the specifically the type of proteins that may be present in a given cell. Now, it is very difficult to quantify using the proteomics technologies how much a given protein may be present, but these are coming in future. I will skip that. I'll skip this. Metabolomics. And metabolomics is in fact is very class. Metabolomics is in fact is a very robust technology which will be able to identify approximately about 4,000 metabolites in a given tissue or a sample, and that is the limit. And metabolomics in fact connects not only the proteomics but also it can connect genomic aspects, because end of the day by doing a reverse analysis of metabolomics into the pathways which are associated upwards, one will be able to identify specific transcriptional events and the translational events that in fact gives rise to this altered metabolomics levels. Especially this becomes very important in studies such as traditional medicine or Ayurveda, depending on the questions which you may ask. There are some examples. Single cell sequencing is becoming very critical, that it is becoming more and more popular these days because assuming and considering the heterogeneity that may be present in a given tissue or a cell, or even in experimental in vitro conditions, no two cells are identical and they would be giving out completely different output or completely different reaction in relation to how these are in fact are operating. And therefore single cell sequencing has become very popular, especially considering the fact that sequencing of genomes or transcriptomes or exomes are becoming cheaper and cheaper, and then scientists find that being able to identify heterogeneity among the cell population by doing single cell sequencing is quite affordable. And there are quite a few papers in the last year which are this year and the last year which have been published to show the power of being able to do single cell sequencing, then being able to derive information in a way including which has been used for the CRISPR-Cas mediated genome targeting, wherein they can identify the sites precisely which is being disrupted by these kind of technologies, apart from being able to identify heterogeneity among the given cell, and single cell sequencing has been quite popular. So we pretty much covered what exactly is entire spectrum of the genome and the kind of technology in about 10 minutes, which I thought is almost impossible. We talked about 5C, ChiaPat, which is a version of Hi-C, and then we have DNA sequence or FAIR sequencing or ATAC sequencing, which identifies precisely the regions within the chromosomes that are transcriptionally poised, or those which in fact bind to the transcription factors or do not bind to the transcription factor, because when the transcription factors are not bound to the DNA, it becomes susceptible to the DNA's digestion. So that is what the DNA sequence or FAIR-seq is all about. We talked about ChIP-seq. BRU-seq is nothing but BRU-UV-seq is nothing but a uridine mediated UV targeted transcription pause and the release of a given cell over a period of time, which is able to identify very early or early transcribing genes within a given cell types. We have computational predictions, whole genome bisulfite sequencing, methylation arrays, we spoke about it, RNA-seq, BRU-seq, eClip, or the RIP-seq is the one which in fact identifies proteins that in fact with the RNA sequences, specific RNA sequences, thereby lead to the discovery of RNA binding proteins. So now I have a task. Let me take another 5-10 minutes if I am not testing your patience, and try to tell you about the genomic basis of Prakriti, some of the experiments which we have done in a collaborative fashion. A program which is being initiated by Principal Scientific Secretary's Office, as Kalpana Joshi has already mentioned, which was initiated by Professor Valyatan, and it was took forward by Professor Bhushan Patwardhan, and we have put together a fantastic team to be able to identify genetic and epigenetic basis of Prakriti. And these include some wonderful traditional Vaidyas coming from FRLHT, from Pune and from Udupi, who worked very closely and very intimately with the researchers from CCMB, Dr. Tangaraj, Dr. Kondaya from Institute of Science, and from us at Manipal University, and Kalpana Joshi, who did both traditional Prakriti assessment as well as the biochemical and molecular aspects of Prakriti program, was also very intimately associated with this program. We had research scholars who have now become doctors, Harish Roti, Govind Raj, Sheikh Nizamuddin. They all worked very hard for 4 to 5 years to be able to complete this program, and now they are working in pharmaceutical industries. So because there is no job which is available in Ayurvedic industry or Ayurvedic institutions or Ayurvedic field. So what a waste of talent, because they are the one who spend 4-5 years with all the technologies which is available at the given time to be able to identify, understand the specific principles of Prakriti. So I will skip this. As we know, features of Kapha, Pitta, Vata, which have been very well delineated, and most of the Vedyas or people who are in this audience, I suppose, are already aware of this, and I skip this. But it is sufficient to say that Prakriti of an individual is in fact is fixed at the time of conception, very similar to what epigenetic and genetic effects, not necessarily what is inherited, but effects are in fact are fixed at the time of conception. And Prakriti is, as you know, has been divided or can be classified into monotypes, dual types and mixed types, and our job is to be able to delineate the molecular basis of Prakriti inheritance. So in order to be able to identify people with the monotypes or the dominant Prakritis, the strategy which was avoided, adopted was of two kinds. One is using simply the expertise of the Vedya, and the second one is to being able to use the AYUSH soft, which can do electronically determine the Prakriti of an individual. So we recruited approximately about 3,500 individuals to the study from the various centers, and then Vedya did the assessment of the Prakriti, and the software also did the assessment of the Prakriti independently without knowing to each other. And subsequently we selected approximately 971 individuals who are supposed to be a dominant Prakriti of a given either Vata, Pitta or Kapha phenotypes. So what this map shows is that the individuals which have been selected from all over the world, different parts of the world, and the distribution of Prakriti that is in fact is seen in the three different centers. The center 1 is Bangalore, and the center 2 is in fact from Pune, and the center 3 is from Udupi, and the distribution of Prakriti among these three centers. And what is most important, and the thing which I am trying to show here, is what is the concordance or the discordant levels between the AYUSH soft as well as the Vedya when they assess the Prakriti of an individual in an independent fashion. So what we find in between the three centers, that is approximately about 20% of the times the software and the Vedyas do not agree, but 80% of the time in fact they agree in relation to the determination of Prakriti by two different methods, and two different methods independently when it is performed. What it came out very clearly, that what is already been known to the Vedyas and the traditional docs, that the body mass index or the BMI, which is in fact is high in those people with Kapha Prakriti as opposed to Vata Prakriti, while it is in fact is kind of in between for Pitta Prakriti individuals, and although which was very well known within the Vedya community, but for us it came as a big observation and finding to show clearly using the validated methods and then providing the experimental evidence and the proof to show that the Kapha Prakriti individuals have high BMI body mass index. So we went on to look at the number of CD markers, what we call it as the immunophenotyping, which provides an extensive information about the type of cells and frequency of the cells, activation of a given cells, and what we find basically that the CD markers CD25, CD56 and the CD14 in fact can distinguish the individuals from whether it's the Kapha, Pitta or Vata Prakriti, because these do belong to the categories of monocytes and activated B cells and natural killer cells, and there is an extensive information is available to tell why these are in fact can be different among the three Prakritis, and because of the short of time we will not be able to explain that at this point, but we can discuss that later. So then what is the genetic basis of the Prakritis? The Dr. Tangaraj group from CCMB basically employed nearly 7,91,000 probes in a microarray platform to be able to identify differences in the single nucleotide polymorphisms across 300 individuals of monotype or the dominant type of Prakriti, and try to identify the variations or unique sequences or unique SNPs that may be present in these individuals. So they also employed Hardy-Weinberg principles. So what is shown here in this Manhattan plot on the right hand side is those which are above the bar, which you see above 4, as in fact are highly significant SNPs that are in fact are present in Vata individuals when compared to Pitta and Kapha individuals. And the list on the left hand side shows the list of SNPs that in fact are maybe present in these individuals specifically in a statistically significant fashion, that is approximately 10 to the power of minus 25, that is highly significant of SNP that is prevalent in the Vata specific individuals. Similarly, for Pitta specific individuals, the line above 4 shows those markers which are highly significantly present in the Pitta Prakriti individuals, and the list on the left hand side shows approximately about 18 different markers which are highly significantly present in the Pitta Prakriti individuals as opposed to Vata and Kapha individuals. And this one is for Kapha individuals, again shows very significant markers, SNPs, which are present in the Kapha individuals as opposed to Vata and Pitta individuals, and the list of about 14 SNPs that have been identified is also delineated and shown here. I know that you may not be able to see it, but this paper has been already been published in the scientific report and it is freely available to read for anybody who may be interested in. The next thing was to be able to look at the epigenetic variation, that is the DNA methylation differences in relation to human dosha prakriti. At the very outset, if you take the entire genome and then look at the extent of CpG methylation or 5-methylcytosine that may be present in these 3 individuals, we find that the Vata individuals have higher, statistically different higher levels of methylation levels or CpG contents in their genome as opposed to Kapha individuals, as has been shown in this picture. So, we used methylation dependent immunoprecipitation technology and used microarray techniques, which I showed you in the beginning, to be able to identify specific methylation regulated regions within the Vata, Pitta and Kapha individuals. And what is shown here again through the volcano plots as well as the Manhattan plots to show the differences in the intra-prakriti analysis as being very minimal to the left hand side in the volcano plot, suggesting that experiments are quite tight and there is not enough variation which we see within the samples which have been used. There is approximately about 120 samples each within each of these groups, and they are quite homogeneous. I won't say completely homogeneous, but they are quite homogeneous because there are some points which are lie within outside the plots, and then using the Manhattan plot one would be able to identify sequences which are differentially regulated among the prakritis by DNA methylation. So, the methylated prakriti associated regions which are associated with the CpG islands within the genome across the 3 prakritis are shown here, and their distribution again through every single chromosome is indicated here, and also considering the fact that they may be present within the promoters or within the G bodies or within the 3' UTRs are also indicated here. All of this information is very important in order to be able to predict and then being able to tell the activation or inactivation of the gene within the location within the given chromosome, and then being able to correlate with the phenotype or prakriti phenotype of an individual. The cluster analysis clearly identify the sequences in comparison with either with Pitta, Vata or Kapha prakritis, where it is indicated here. I will skip this, and what is shown here is the association of BMI with the prakriti, as we said that Vata prakriti have high BMI, and then that it also is providing us with the genomic information, epigenetically regulated information to identify specific regions within the chromosome which are not only associated with Vata prakriti but also with the people with high BMI. And the most important thing is to be able to validate some of these signatures. Without validation, whatever the results which are coming from high throughput analysis is simply cannot be relied upon. So, we did hundreds of validations of the genes, and I do not have time to go show you all of that. I will give you just one example and then I will conclude my talk. One of the gene which we looked at was LHX1, which is in fact the limb domain containing transcription factor, which we find that it is very highly methylated in Vata prakriti as opposed to the Kapha prakriti, as it is indicated here. When we analyze individual sequences within the promoter sequences of LHX1, you can see that Vata Prakriti genomes are highly methylated within the marked region as opposed to the Kapha Prakriti, suggesting that these gene regulation in fact is imparted by the CPG site specific methylation and their activity in relation to gene expression. Similarly, we also looked into a cadherin superfamily protein CGS22, which is in fact is kapha methylated and also associated with the BMI as it is indicated here. With the high BMI it is methylated, and with the low BMI which is less methylated. And these very data gave us an indication of those sequences within the human genome which are differentially epigenetically regulated, and these are in fact are providing us with the potential opportunity to look at how the epigenetically regulated DNA methylase signatures may contribute to the phenotype of BMI which is associated with the individuals. These also studies gave us quite a bit of information about copy number variations within the Prakritis, and these studies are currently ongoing, and more importantly we are also in the process of deciphering whether there is an inheritance pattern in the Prakriti in a given family or in a given individual inherited through the families. We identified number of microRNAs which are associated with the methylation, which is currently the studies which are ongoing, and these are some of the studies. And the gut microbiome which has been done by Dakkan et al. Group, where they have shown Prakriti specific microbiome population, but there are some issues which are associated with that, with the 3 studies which have been done which do not show complete coherence among the microbiome population which is in fact is present between the 3 Prakriti individuals, except to the fact that there are a number of variables which are associated with that. You have a diet, you have lifestyle, you have region. In fact, the studies done by Chauhan et al. Would show there is a completely different set of microbiome which are present in the North and South Indian population as opposed to the population which is studied for the Western population. So the studies which encompasses pan-India sampling and then hundreds and thousands of individuals would in fact be necessary to be able to, as has been proposed earlier in the talk before, to be able to decipher accurately the role of microbiome in relation to Prakriti. So I skip that. Many of these papers have been published already. Once again, I thank to our dear collaborators, without whose help and cooperation, the knowledge which we have learned, this work would not have been possible. And thank you very much for your patience.
There can be stupid questions, but intelligent answers.
Not coming from you.
Indian population have a massive deficiency of vitamin B12 and folate also. And as a consequence, acetylnosyl methionine levels are also not done but expected to be. We know that homocysteine levels are very high in Indian population in general. So what I wonder is, when we are studying methylation, and methylation itself is a very but I would also say that histone deacetylase, and those are also important things, because the data are showing that gallic acid and some of the polyphenols and all are affecting histidine deacetylase. So first question is, you focused on methylase, fine, but what about histone deacetylase and histone regulation of DNA expression?
Yeah, very good question, sir, very good question. The methylation which I showed you, some of the results are in fact governed by the 1-carbon metabolism, in which homocysteine is a product which is coming out of that. And again we see the difference between kapha, pitta and vata, where the difference in methylation. Coming back to histone modifications, especially histone methylation, histone acetylation, histone phosphorylation, histone ubiquitination. And very early in my talk on the omics technologies, I talked about 2C, 3C, 4C, 5C, high C, chia pet, and also DNA's footprinting, and those are the kind of technologies would give us a genome-wide map of these post-transcriptional modification that we can see in the core elements which are associated with the DNA. If the government is willing to fund us, we will be very happy to do it, and we will not stop trying, so we will try.
No, my question was confounding variables. When you apply, okay, we think that we are studying Prakriti. Prakriti, as per Ayurvedic definition, is something which is fixed at the birth. The entire biology is showing that nothing is fixed. You can have gene expressions, modifying the phenotype, modifying the metabolism, all that. We know that by now. So when the background noise is there,
Yes, sir. Yes, sir.
how do you control the background
Yes, sir,
noise?
precisely, sir. I don't know whether Dr. Shauche is here. The word he used two words, right, which is also used in the genomic area. One is called core microbiome used. I use core changes. Then we have a pan changes. The core changes, so-called housekeeping. I won't say precisely housekeeping. They would more or less remain the same, but they are the small proportion. And the pan changes, the pan epigenetic genetic changes, they are more frequently seen across the genomes. And I think especially for nuclear changes, nuclear architecture, nuclear post-translational modifications, the core would probably remain the same, and with the pan changes which one would look at, and that would probably have a significant or impact on the architecture or the effect of these post-translational modifications.
So when you are planning the future studies,
Sir.
is there because Bhushan and myself have been discussing it for many years, that what we call Pitta Prakop Davyas, Vata Prakop Davyas, Kapha Prakop Davyas, Kapha Shaman paths, we know all this.
Yes, sir.
And whether we can have a gene expression study, genomic studies or metabolic studies in relation to utilizing these agents and studying the markers and comparing it with the phenotypic changes described also. So the Pitta Prakriti will not tolerate the sunlight well. So we need to evolve some tests, simple tests, rather than this questionnaire of Prakriti.
Absolutely, sir. Absolutely. And I think you are talking about the vision of what we should see, and in 5 years, 10 years down the line, and this is what is important. And questionnaire is, yes, but as you know, it is a little bit of subjective, but it is nothing like having a specific markers with the range to be able to tell Vata, Pitta, Kapha, Prakriti, associated conditions and the diseases with the diseases, and that should be the vision for next 5 to 10 years, and this needs to be driven and this needs to be implemented. And that is where we have the Indian science, our own science.
Thank you.
Thank you.